My name is Evan Lowe, and our group chose the article "The absence of specific heat shock protein lead to abnormal aggregation and compromise autophagic clearance of mutant huntingtin proteins." The purpose of this article was to look into certain proteins that have been led to aggregation and their correlation between Huntington disease and the creation of the disease. Huntington disease, we need to look at what Huntington disease is first. Huntington disease is an inherited condition in which nerve cells in the brain breaks down over time. And currently, right now, there are fewer than 200,000 U.S. cases per year. The cells that this article focuses on are the certain nuclear exchange factors that are located within the chaperones. These exchange factors are known as nucleotide exchange factors, and their functions are to exchange ADP with ATP. SSE1 and FSE1 are located in the HSP70 chaperone, which function is to help facilitate protein folding. HSP70 chaperones are a highly abundant ATPase that is able to bind to the hydrophobic region of substrate, which helps facilitate protein folding. Most of the function of HSP70 chaperones are SSE1 and FSE1. The paper that I chose was the autophagic degradation of HTT103QP, which is compromised by the SSE1 and FSE1 mutants. During this function, we looked at how these two factors are able to not degrade within the protein. During this experiment, the SSE1 and FSE1 mutant cells have been placed with the HTT10QP expression, which is then induced in a galactose medium for 16 hours, and then glucose and DNA synthesase inhibitor, which is known as hydroxyurea, was added to the medium to shut off the expression and prevent cell division. By doing so, we are able to see how the mutant SSE1 and the FSE1 mutant cells can prevent their aggregation and their deaggregation of its cells due to its mutancy. The results of the experiment show that after a 90-minute incubation, the wild type, which is the control, had 26% left containing the HTT103 QP aggregates, but then 72% of the SSE1 cells contained the aggregates. In another experiment, after 120 minutes incubation, 53% of the FES1 cells shows a HTT103 QP aggregates compared to the 19% of the wild type cells, which helps prove that these two factors, if mutated and put into the cells, will block degradation of this mutant proteins. In our conclusion, the researchers talked about how the SSE1 and FSE1 mutant cells shows a larger amount of aggregation between each cell and exhibit a significant change in the IB formation. IB formation are also known as interbrotic inclusion body formation, which is an active cytoprotective process where the misfolded protein species are able to fold and refold over and over again. Their findings suggest that the chaperone network is able to regulate the mutant Huntington disease by preventing aggregation of the misfolded proteins.